Okay, this is section 2.5 of the AP Biology course and exam description, membrane permeability. And I know I talked about a lot about this, so I'll probably get through it a little bit more quick. It's section 5.2 of your textbook. And we're going to look at how the structure of membranes influences its selective permeability. And this animation shows you the fluidity of the membrane and some components of the membrane. I'm being attacked by flies. And how permeable the membrane is to different substances. So if we just look at oxygen, for example. Oh, look at that's. I'll show you that. Let's look at that again. Oxygen goes right through the membrane from high to low. So that's simple diffusion. Carbon dioxide, same thing. It's going to go right through the membrane, carbon dioxide. Let's look at glucose. Glucose is kind of big. It's also kind of polar. So glucose needs a transporter to get through. It's still going high to low, facilitated diffusion. All right, potassium has a charge, so let's see what happens with potassium. Okay, ion channels. You need to have an ion channel for ions, things with a charge to pass through. Same thing should be for sodium. Oh, look at that one. 3 sodium out, 2 potassium in. 3 sodium out, 2 potassium in. What they're not showing you is that there's going to be requiring ATP. So that pump is going to be active transport. It's going to require ATP. Whereas the ion channel for this potassium was high to low, didn't require. It's not a pump. Okay, water. Let's look at water. Oh, I didn't click water, I don't think. All right, so look, some water goes right through the membrane, but more goes, or it doesn't show you more, but more is going to go through. So there's a couple different ways little bits of water can get through simple diffusion because it's small enough, but most of it's going to go through the aquaporin. And then enzymes, let's see what we have for enzymes here. Oh, that's cool. Enzymes are big molecules, big proteins. So to get through the membrane, it's going to have to use exocytosis to get it out of the membrane. It's going to be exocytosis by active transport, secondary active transport. So that's a good little overview. I like that animation. That's linked in your document. It's linked in your document, I believe the first one there. Okay. So I'm going to come back to, this is a free response question. I'm going to come back to it in a minute here. We'll do that one at the end, just like I did last time. So this kind of just shows you some increasing permeability. So we already know CO2 and O2 have high amounts of permeability. Water, less so. And then you don't need to memorize these, really, not most of them anyways, but you should know things that are charged and things that are big and polar. So sucrose, glucose, fructose. Glucose is a little bit more permeable than sucrose based on size, but not by much. Both of those are going to be less permeable. Something like an amino acid. So a lot of these require specific proteins in the membrane to get them through. CO2 and O2, they get through pretty easily. Ammonia, water, a little bit less easier, but still pretty easily. Something like sucrose, that's not going to get through the membrane very easily. Too big, too polar. All right, let's see what we have here. Something like, what's interesting is that even if it's pretty big, because of the membrane being, the things that are soluble in fats, like steroid hormones, they can go through the membrane pretty easily. Um, Whereas something like glucose, proteins, and are going to be more harder to get through because of their size. Lipids it depends on the lipid. If it's a big lipid, it's not going to be able to get through. If it's something small like a steroid hormone, it can get through. So that's the biggest difference there. Size, solubility based on polarity. Things that are nonpolar are more likely to go through, and things that are charged or are polar are not likely to go through. All right. So lipid soluble molecules, it depends, especially if they're pretty small, are going to be able to pass right through. All right, so this is just a graphic I found that had a good summary. So they're permeable, slightly permeable to water, lipid soluble things, gases, things that are small. So highly dependent on size. The smaller, the faster it gets through, and its ability to dissolve in lipids. If it's able to dissolve in lipids, more likely to get through. But they're not going to be permeable to things that are big, polar, or charged. Okay. That's kind of just showing us the same. This is basically the same look at everything here. All right. And then the last thing on here is going to be talking about the cell wall. And I just want to talk a little bit about differences in cell wall between different organisms. So really, the only group of organisms that doesn't have some sort of cell wall are animal cells and some protists. But bacteria have a cell wall, and fungi do, and plants, of course, do. But just make sure you understand that plants are not the only types of organisms that have cell walls. Just to kind of talk briefly about different bacteria, both bacteria, whether it's gram positive or gram negative, we're going to probably talk about this at some other point in more detail, have their cell wall is mostly made up of something called peptidoglycan. And this is actually a target of when we talked about targets of antibiotics, one time we talked about the target being the ribosome. And in some cases, especially like in something like I think it's penicillin, will target the peptidoglycan layer of the microbe. And so some of them have a very thick peptidoglycan layer. Some of them have a thinner peptidoglycan layer, and then on top of it they have the lipopolysaccharide layer, and then they have their other membrane. And then, so that's how it's just two major groups, huge major groups of bacteria. One is being gram positive and one is gram negative. One of them stains purple because this peptidoglycan holds onto the stain very well. One of them, you can wash that stain very quickly out because you would counter stain it red. So that's where they get their name from the person that developed this stain, gram positive and gram negative. All right. And then for fungi, the big thing I want you to remember about the fungal cell wall is that it's made up of chitin, okay? Just like insect exoskeleton, very tough. So mushrooms, they have kind of a polysaccharide chitinous cell wall component to them. And then, of course, you know that plant cell walls are made up of cellulose. Okay, most of their cell wall is made up of cellulose microfibrils. They have a lot of other things in them, some things that glycans and pectins that are sugar-like materials that kind of hold the whole thing together. And like plants that have that are like green stems, that's going to have a big layer of primary cell wall. But then as the plant ages, it thickens up and it becomes more like a woody stem, and that's a secondary cell wall. Okay, so I think that was it for some of the things about the membrane permeability. Like I said, it's a lot just like the previous. So I've just kind of taken these one day at a time when I'm making them, and I didn't really realize that this all kind of fits in with what we were talking about yesterday, but that's okay. So the, I want you to think about this one for a second. This one is challenging right now for you, but remember what we've talked about so far. So estrogens are small, hydrophobic lipid hormones. So, right off the bat, I'm thinking estrogen is going to be able to get across the membrane pretty easily, okay? Because it's small and it's hydrophobic. So, those are two big considerations, that this is going to dissolve right through the membrane pretty easily. What estrogens can do, they're like growth factors. So they're involved in cell signaling. They're hormones. They tell the cells to do things. And one of the things that they do is promote cell division and development in general. So, and it tells you right here that estrogens passively diffuse through the membrane and bind to their receptor proteins in the cytoplasm of the target cells. So it's telling you, it's giving you a lot of information here that you can use to answer this. Estrogens get across the membrane, but their receptor is inside of the cytoplasm of the target cells. Okay? And one characteristic of the plasma membrane that allows, this is going, I'm going to find a space to write, because remember, we want to write in a paragraph form. We don't want to just, well, we can't. We have to make sure that we write it in complete sentences. So it's asking to describe one characteristic. One character of the plasma membrane that allows estrogens to passively cross the membrane is that a large portion of the membrane is made up of non-polar fatty acid tails of the phospholipids. Because the membrane behaves like a fluid, when the non-polar steroid, I'm going to change that to lipid hormone, estrogen, contacts the membrane, it is able to passively cross the membrane. Okay. And then it's telling us, in a laboratory experiment, a researcher generates antibodies that bind to purified estrogen receptors extracted from cells. So there's an antibody that is able to bind to the estrogen receptors. Okay? So what would happen then is estrogen normally promotes cell growth, and if you have like a breast cancer tumor, estrogen is, in some cases, estrogen will promote the growth of that tumor. So what the theory behind this is, if you can get that antibody to block that receptor, then estrogen can't come in and promote that, send the signal to promote that cell growth. Okay, so in theory that sounds to be a really great, and it does work in some cancers. But the researcher finds out that is giving that medication, assuming you're taking that medication either through IV or it's a chemotherapy or by pill, they find out that that treatment is ineffective. So they want you to explain the ineffectiveness of the antibodies for treating estrogen-dependent cancers. Okay. So let me think about this for a second. Estrogen-dependent cancers require that estrogen be estrogen promotes, I'm thinking on the fly here, can promote cell growth by binding to the receptor, to the estrogen receptor inside the cell. If the antibody can bind that receptor instead, it can help stop estrogen dependent growth. However, we're talking about membrane permeability here. Antibodies, this is just something you have to know, antibodies are large protein molecules. Large protein molecules will not be able to enter the cell and pass through the lipid bilayer. Therefore, without binding the receptor, they will not be able to stop estrogen from promoting, they will not be able to block estrogen from promoting cell growth. So that's what I would say there, okay? Again, you want to write in complete paragraphs or complete sentences. You want to pretty much just get to the point, answer the question. Make sure you read the question, because that's important, because you get good information from there that, if you didn't know, I want you to know that estrogen is lipid, and it's a smaller lipid molecule that can pass through the membrane. I mean, it's a big molecule, but because of its being nonpolar, it's able to get through. And I want you to know that estrogen is something that promotes cell growth. It's a hormone, and it needs, and then hormones need receptors, but there is a lot of that information that's given here as well. So you have a lot of information. You just need to make sure that you put it together. Okay, so I hope that was helpful in answering that extended response question, for that free response question for you guys.